Satellos is a clinical stage drug development company focused on restoring natural muscle repair and regeneration in degenerative muscle diseases. CEO Frank Gleason and CFO Elizabeth Williams are here to provide an update and discuss the recent data from the Phase 1b open label trial of SAT 3247 for Duchenne muscular dystrophy. I'm Martin Gagel with Market Radius Research. Please remember this is neither recommendation nor investment advice. We're here to learn about the company. Frank, Liz, welcome back.
Thank you so much, Martin. It's great to be back on your program and to speak with your audience. And we're delighted there are so many that are tuning in or will do so subsequently. We look forward to a dialogue.
All right. I'm really looking forward to digging into the data from the Phase 1b study. But first, go ahead with your presentation. And then we've got lots of questions already in from the audience, and I've got a bunch queued up for myself. So take it away.
Okay, perfect. So we'll walk through a refresher once we get through the legal disclaimers, then remind you that we will be making forward-looking statements and we're a publicly traded company. We'll give you a refresher on Satellos. We'll walk through some preclinical data to reorient the audience, then our clinical data, and then we'll talk about the forward plan. And we'll be delighted to deal with questions as we go along. So as Martin said in the outset, our focus is the regeneration of muscle with the idea and the intention of restoring function for individuals who live with degenerative muscle conditions. Our science is steeped in a very deep history of understanding muscle and muscle regeneration and built on publications in the most elite journals where we have made what we believe are disruptive discoveries and certainly highly differentiated thinking about the whole area of muscle and bringing muscle back. We believe that our drug 3247 has the potential to be disease modifying in Duchenne muscular dystrophy and potentially other conditions. And DMD in particular is a situation where there are no disease modifying treatments. Our pill is designed to be safe and well tolerated and essentially taken on a daily basis for life. It's intended to increase function versus delaying the loss of function, which is what many treatments attempt to do. And there are applications in multiple degenerative conditions through to aging. And as Martin pointed out, we're a clinical stage company. I'd like to add to that we have a strong balance sheet. We'll talk about financing later in the discussion. We will have a separate discussion about our flow of milestones and potential reporting, but we have what we think is a very robust cadence and schedule of upcoming events that we'll be able to talk about publicly as we move along. We have a great team, and I think many of you on this call already know that we have an elite institutional investor base of very high-end professional biotech investment funds that are US-based, and they are long on the stock and very supportive of what we're doing. And what we're doing is trying to find a way to create a better life for young people such as Charlie. Charlie is a real person who is 7 years old, living with Duchenne. And we think about Charlie and other children and adolescents and young men living with this disease every day of our lives that we're working on trying to develop a treatment for them that helps them live a more easy life and hopefully brings back some functional capability that they start losing at too young an age. And what does that mean? So the next slide describes what the Duchenne journey is like for individuals living with Duchenne. And you may have seen me use this slide before. What it depicts is the progression, the constant ongoing progression of functional loss. it also depicts something very interesting that maybe has been hiding in plain sight for far too long. But early in life, children that are born with Duchenne muscular dystrophy actually grow, have muscle function, gain muscle, and have strength. And often, until a child is 4 or 5 years of age, their parents, family members, friends don't notice that there might be something that isn't quite the way it should be in terms of their young son. And that not only surprises us that this hasn't been focused on before, but it basically, kind of puts a line through the argument that muscle can't be functional if it doesn't have dystrophin. These children are born without dystrophin. They start to grow. They start to build muscle. They start to function. They walk. They can play. They can throw a football and do all sorts of other things. And then at a point in their lives, usually in that 5 to 9-year-old period, they begin to experience severe loss of function. And before they are teenagers, generally speaking, they become unable to walk, and they have to move into a wheelchair. And then over the next 2 decades, they lose more and more function. Now, we focused on why they lose the function. They lose the function because they physically lose muscle. It's not that muscle is damaged and weakened. That is true. But it's that they lose muscle. And the question we've wanted to answer is, why do they lose the muscle? Because damage doesn't equate to loss. And what we discovered is they lose muscle because it's not regenerating the way that it's supposed to. And that's what's depicted on this slide. And I think some of you that have followed us have seen these slides before. But just to put it in context, in Duchenne muscular dystrophy, unlike in a situation with a healthy system, muscle doesn't repair itself. So looking at this fiber, you can see we've depicted the fiber becoming damaged. Now, in muscle fiber, there are muscle stem cells. Those muscle stem cells basically are asleep. They're quiescent cells. In response to damage, they spring into action. That means they go into mitosis, the basic biological process of cell division. They only do that in response to damage. So cell gets damaged, muscle cell gets damaged, mitosis occurs in the stem cell. And then the stem cell has to divide. Now, dystrophin, we learned, provides a particular and unique signal inside the stem cell to tell that cell how to divide, to create both a stem cell and a new muscle cell, a progenitor cell. Without dystrophin in the stem cell, that doesn't happen efficiently or at all. Instead, more stem cells get made, and you wind up essentially with too many stem cells. That is exactly what is seen on the right-hand side in those bar charts. The green represent stem cells, and the purple represent new muscle cells, progenitor cells. And clinicians that treat Duchenne patients have known as far back as the 1980s, in looking at biopsy material of human beings living with muscular dystrophy, that they have what seem to be too many stem cells in their muscle and not enough new muscle cells being created. Now, they didn't have the tools or the understanding to truly know what they were looking at and the significance of what they were looking at. But we now know that that's a huge clue that the body is trying to repair itself and failing, and that the stem cells are still there. So that's where we come in. Knowing that, which is our discovery of why this is happening, we have designed a pill specifically to fix that problem. And the way we fix the problem is to replace the signal. Because it's a signal, that means proteins are involved with talking to one another, with interacting with one another. This is basic biochemistry. Proteins interacting with other proteins. That's what goes on in our bodies all the time, in every part of our body. And in the case of muscle stem cells, we found another way to provide that signal that's missing from dystrophin. We're not putting dystrophin into the stem cell. We're working with what the stem cell has. We're not adding anything to the body except a pill. The individual takes a pill, and the pill interacts with a protein called AAK1. AAK1 has the capacity, we discovered, to replace the missing signal. And when it does that, what happens is the cell then knows how to divide properly. It more or less stands up. So it now orients itself physically, geospatially, into this aspect where it's perpendicular. It's now at a 90-degree angle to the muscle fiber, and it divides to create two different cells. A stem cell, which goes back into the fiber for future use, goes back to sleep, and then on the other side, it creates a progenitor cell, the muscle cell. That cell will create a legion of up to 2 to the 12th new cells. I think that's something like 4,100 cells. Literally, it creates thousands of new cells that then home into the damage and repair it. And we think of this as repairing from the inside. It's like we're fixing the muscle from the inside. And that brings back muscle function. So next slide, please. So just before we get into data, just a quick recap of who's doing what in the space. So there are other players that are listed on the left-hand side, but these are some of the big names that you're undoubtedly familiar with. And the most common approach to treating Duchenne muscular dystrophy that is being developed is genetic approach. And that's either through gene therapy by DNA or exon skipping by RNA. So the two aspects of genetics. And the aim of putting dystrophin back into the body by one of these two means is to stabilize the muscle fiber, to slow the progression of the disease, and hopefully that provides a benefit to the individual. These treatments often have limitations related to specific exons or immune backgrounds of the individual. Our approach is a daily pill, no immune issues, no exon limitations, but the key difference is our goal is to regenerate muscle, to reverse disease progression. So slow it versus reverse it. And if we are right about that, we should see increased strength or increased function as an early sign that our treatment is being effective. And then this creates the possibility for a drug that could be a standalone therapy or complementary to other approaches. That's what we're working on. So we'll walk through some data. So I just want to revisit canine data that we shared some time ago, because this has come up in questions to us in the context of human data that we've released. A reminder, we conducted a 4-month study in canines with DMD. We treated 2 animals for 4 months in total, and we measured strength at 2 months and 4 months. And when we compared that to natural history of healthy animals or of DMD animals, what we see is that we brought these animals that we treated, that were actually much older at 9 months to start, back to the level of healthy animals. In other words, rapidly, early on in the process, we showed a benefit in strength, which we believe offers promise for a functional benefit to be an early signal in clinical trials. And secondly, what we saw when we looked at histology, which means when we looked at biopsy material on the next slide, we repaired the muscle. So you can see here on the left, untreated muscle at the beginning of the study from one of the animals. In the middle, the same animal after 4 months of treatment. And on the right, a healthy animal not in the study used as a control. And you can see a dramatic difference, I think you would agree, between the untreated and the treated, and that the treated looks very similar to the healthy. And what has changed across these images is that in the untreated biopsy material, what you're looking at is muscle that's breaking down and being destroyed. And the massive amount of red staining that you see, other than the lines, which is connective tissue, but the blocks, sort of globs of red, are just a color coding for an immune factor called immunoglobulin G, which is a signifier that the immune system has entered into the muscle fiber and is beginning to devour the damaged muscle tissue as a way to cleanse the system. But the problem is that muscle doesn't come back. What we see in the middle is that when we treat, the muscle is coming back. And that's the nice black images. Those are the fibers, the sort of cables that you're looking at as though you're looking from the top down. And the green is the laminin that is connective around those fibers that stains to show you the boundaries of the individual fibers. So that then, of course, looks very much like a healthy situation. And then on the next slide, we show this in greater detail. So now what we're doing on this slide on the left is showing you that we can actually, through molecular means, count all these cells, count the number of IgG cells versus new forming fibers, and get a ratio of new muscle cells to dying muscle cells. That's what the left is all about. And that requires a biopsy. We have to have the tissue to do that. And on the right-hand side is sort of a measure of the knock-on effect from our drug on the immune system when IgG starts to go down. It has an effect on what IgG affects, which are all kinds of cytokines, which are elements of the immune system that actually do the kind of dirty work of, breaking down and cleaning up damaged and dying cells and getting them out of the system. So there's a lot going on here, and a lot that we will have the ability to use as potential markers in clinical development. So let's go on to the clinical development. So the first thing that we did was a phase 1 trial in healthy volunteers. Now, we designed our phase 1 trial to have 2 components. Healthy volunteer component, 1A, we called it, and DMD adults, 1B, we called it. And the purpose in both of these components was primarily safety and understanding the exposure of the drug in the body, what is called PK, pharmacokinetics. So in the healthies, we followed a prescribed format that the regulators insist on seeing. You first do what's called a SAD study, not because it's sad, but because it's a single ascending dose. And then you do a MAD study, which is a multiple ascending dose. And you also look to see if your drug is affected by food. So you have one cohort that is given a meal prior to taking the drug, and then you look at the PK and the safety with food on board. So we did 72 patients, and I'll tell you about the data in a moment. And then once that study was complete, and once we had enough evidence of the safety of that study, the regulators in Australia allowed us to begin to recruit adult DMD patients. Now, why did we do adults, not children or some other group? Because at that time, when we were doing this study in the fourth quarter of 2024, we only had tox data for adults. So in order to move into younger age groups, regulators require that you do different levels of toxicology studies. The younger you go, the greater the demands, and the more types of studies need to be done. And so, working with what we had, we thought, here's a very interesting way for us to get a sense, is our drug affected when the individual is living with Duchenne and is on steroids? Because we couldn't do that in healthy volunteers. And while we were doing this safety study in the adults, we thought, to be fair to us, to get more data, and to be fair to them so that they could contribute as much as possible, because that's what they want to do. And often this is what patients want to do when they volunteer to be part of studies of this nature. We thought we would look at some exploratory markers to learn more and see what we could see. So if we move on, what we saw in safety in the 1A, single ascending dose, at doses from 10 milligrams a day to 400 milligrams a day, basically totally safe. At 400 milligrams, which is about 8 times what we will dose humans at in what we think will be our therapeutic dose, we saw a little bit of nausea in one person and a little bit of pain, abdominal pain in another person, probably because they had to take 8 pills to get to 400 milligrams. In the multiple ascending dose, at doses from 60, which is approximately where we will likely start working in our phase 2 program, up to 240, which is several times higher, essentially we saw a very, very clean profile. We reported that we saw a tiny little bit of elevation of a couple of markers of liver. They're called transaminases, ALT and AST, and these are significantly below any concern of a clinical nature. And then we saw a little bit of, gastric upset, tummy upset at the higher doses, and not much else really. This is about as clean as you can possibly get a safety trial to look. And then we also looked at the exposure. So how does the drug behave in the bloodstream? How long does it get? Does it stay in the bloodstream? And how much of it gets broken down and is detectable in the bloodstream? You want to see that. So that creates a curve. And what we were looking at, because we had set some objectives, was what happens over a 24-hour period. Now, we did look longer, so we know what happens beyond 24 hours. But what we really wanted to see is, does our drug at what we think will be therapeutic doses, does it clear within 24 hours? And for our mechanism of action, based on all the studies we did internally, that's important. We concluded that was important. And what you can see very clearly on the graph on the left, whether you're an expert in drug metabolism or not, you can see very clearly that these graphs peak very quickly within 2 to 4 hours, and then they go into decline. And the red, the purple, the green, that drug is all gone by 24 hours. And even at the highest doses, the drug is basically leaving the system at 24 hours. So it's perfect for what we wanted. Then we said, okay, how does that look compared to where we think the dose needs to be to hit the target? So you can see a dotted line called AC1 IC50 for 3247. So 3247 is the drug. The IC50 is basically a measurement of inhibiting the drug. So IC50 means inhibition of the concentration at 50%. So you want your drug to inhibit your target at a certain concentration, i.e., you want to know that you're inhibiting at least half of the target. And we wanted to do that for 6 to 8 hours. That's what we want to do. But we also have an upper limit on it because we want maximum safety. Because in these children, they are on so many drugs that have so many side effects. We think this will be a huge competitive attraction if we have a safe, tolerable drug. And so we set a very high bar, which is a 10 times safety window, which is basically called NOAEL for not observable effect level. And you can see that within that, 2 of our lines, well, 3 of them, the red line wouldn't get above the IC50, but the purple and the green line achieve this beautifully. And so we basically have met our objectives for a drug that meets all the targets that we spent about 3 years trying to figure out. So that's beautiful. Then we went into the adults. And in the adults, just to remind you, this was a safety and a PK study. So we're looking at safety and we're looking at the exposure levels first and foremost. And we also explored some exploratory measures. 5 patients entered the study. They were aged 20 to 27. All of those patients have been on steroids basically their whole lives and continued to take steroids that were prescribed to them during our study. This is important because when we go into phase 2 development in other age groups, they will be on steroids. And we wanted to make sure steroids did not somehow affect our drug in ways that we haven't been able to predict. They were on drug over a 28-day period. They received drug each weekday, Monday to Friday, for 4 weeks. This will be our dosing pattern in clinical development, Monday to Friday, weekends off. They received a 60 milligram dose, which we predict will be our therapeutic dose, most likely. And our primary endpoints were met. The drug was safe and well tolerated. I think, frankly, we saw even less than we saw in the healthy individuals, and the PK profile was unchanged. So the steroids did not affect the PK profile. So that basically means we met the objectives of our study. And then on the exploratory measures, here are the highlights of the data. And basically, this is the data. So we looked at a number of different things, as I showed you on a previous slide. We looked at grip strength, which is basically asking these individuals to squeeze a kind of gripper, which has, two poles on it, and you squeeze them together and measure the force. Pinch strength, same kind of idea. Lung function, so, that spirometry, where the nurse gives you a tube, you have to either, blow into that or draw breath. Serum markers, so a blood draw, and upper body effort, which I'll explain in a moment. One thing absolutely jumped out at us, and it was massive. We thought massive. Grip strength. We saw a doubling of grip strength in these individuals. And many of them, they start with very, very low grip strength. At this age, these young men have very little muscle left and very little strength. And so to see a significant increase over a 28-day period just blew our minds. Now, we saw this increase in 3 of the 5 patients in both hands. So the statistics are driven by 3 individuals who showed very significant changes in both hands, and changes that aren't due to sampling error. And then the other 2 were stable, which means essentially they didn't change. They didn't get worse, but they didn't really change. When we looked at pinch strength, there were no discernible trends. Essentially, it was all kind of flat. When we looked at lung function, 4 patients averaged a 5% increase, and it was reasonably consistent across the 4 of them. The 5th patient suffered an injury and wasn't able to conduct the 28-day the breathing test at 28 days. So that patient did not. We don't have data for that patient. Nothing to do with the drug. These are sick people, and they often have other complications. We looked at serum markers. We looked at serum markers on day 1 and day 15. It was very, very difficult to do blood draws on individuals of this age with Duchenne. Often getting a vein was a real challenge. So we did not want to do too many blood draws with them. So we picked day 1 and day 15 because that coincided with what we needed to do all of our PK work. What did actually surprise us is how low the levels of cytokines and CK were in these patients to start with. Now, one very big reason for that is that they've been on steroids their whole life. And our dogs weren't on steroids. So when we saw the cytokines in dogs, it probably would be predictable that in humans you would see lower levels of cytokines because they've been on steroids for so long. But the other big factor is they don't have very much muscle left. So we didn't see very much in the serum markers. We will continue to look at this as the study continues in a long-term follow-up. But it's not because we're diminishing the importance of serum markers or the desire to have serum markers. I think it just reflects the reality of the disease as people get older. And then the upper body effort was an experimental wrist monitor that we decided we would try in this study. And it allowed for a measurement. It was worn for the entire period of the study, and it allowed for a constant sort of measurement of the ability of the individual to move one arm. Very different concept than grip strength or pinch strength. And as of yet, there are no trends across the patients that are discernible to us or advisors that we've been working with. So we'll continue to look at this over the course of the follow-up study and continue to monitor. But what really jumped out was the grip strength, and this is consistent with what we've seen over and over again in our preclinical models. We always see there's an improvement in strength, and it's very quick. And just to put it in context, this is what it looks like graphically. So the Satellos data is on the left. For some reason in the clinical site we worked with, they referred to it as the non-dominant hand and the dominant hand, as opposed to left and right. But it's the two hands. I repeated in the little panel the data that we talked about on the previous slide, but here it is graphically set to a baseline of 0. And on the right, what you're looking at is data that has been publicly shared by an exon skipping company. So if you want to do a Google search of the image, I'm sure you'll find it, where they looked at 8-year-old patients, 11 of them, for 48 weeks. And what they saw was a 1 kilogram average increase in grip strength, left and right hand revealed here. So that's 48 weeks. Our study was 28 days. Much older people. So, this is why to us, this functional data just jumped off the page, and it certainly excited us. We'll just continue, Liz. Okay, so we'll talk a little bit about the clinical plan going forward. I'll try to be as clear as possible on this because I think it reveals a lot going on. So it could be that it's a little confusing. We completed the phase 1A at the top. We presented that at the Muscular Dystrophy Association in March 2025. We've now completed the 28 days of the phase 1B. We did a top-line reporting of that data at Cure Duchenne 2025. That was a big deal for us because that was the first time that we have been at a Cure Duchenne meeting. We were invited by Cure Duchenne to come to that meeting. And we believe it's because they have started to see the progress that we're making, and because their families and their supporters are very interested in learning more about Satellos. What we didn't realize was that it wasn't an open meeting so that people could see the presentation online. So I do want to apologize to everybody for that. We did not understand those ground rules at the beginning. So we will make sure that this presentation goes on our website, and so you'll be able to see the data that we've just disclosed. And we are now going to move into a long-term follow-up on that phase 1B study. So the patients that have enrolled and been treated for 28 days, we're inviting them to continue on drug for 11 months longer. Now, this is actually a pretty big deal because this required us to get regulatory approval in Australia for 11 more months of dosing. And they would only give us that approval if they thought this drug would be safe, because that's critical. So this gives us now 11 additional months to look at safety, to look at all of those exploratory functional factors, and to have a steady stream of data that we'll be able to read out. And we'll show you on the next slide in a moment what that could look like. We are also, in parallel, preparing to initiate our phase 2 program. Now, this is an entirely different program in a very different population. We are going to do a global study. So this study will be in North America, in Europe, and also in Australia, where we will look at pediatric patients. So these are children younger than the age of 12. And in this case, pediatric means that they've not yet gone through puberty. So they'll be younger children. We are going to do 51, an N of 51. If the regulators approve all of this, this is our plan. This will be a randomized study. It will be placebo-controlled, which will mean one group will not be receiving drug. We will have two different dosing groups. So that means two dosing groups will be receiving drug. So we will have a comparator. We will be able to compare untreated to treated. The study we did in the adults was what's called open label. There was no treatment. There was no untreated group to compare to. And the reason we're doing this, and the level of dosing or the length of dosing will be 3 months. Now, I'll tell you why we picked all of this. We decided on this because we're so excited, number one, by the data we started to see in the adults, that we wanted to accelerate our overall plan to get this drug in front of regulators and to generate the data that could lead either to a way to get approval or could lead to M&A activity of a significant nature. And so we thought we needed to get into the target population of younger children as fast as we could, and we needed to do a placebo-controlled study so that there would not be any doubt that if we were seeing an effect, it was real and not due to some kind of effect related to effort or perceived effort or trickery or not having all the data or whatever happens when you don't have a placebo group. We will look at a wide range of endpoints related to safety, muscle function, imaging of muscle, biopsy of muscle, serum markers, all those kinds of things. And we picked 3 months for a couple of reasons. But one of them is that in the late 1990s, steroids received approval for treatment and became standard of care in Duchenne muscular dystrophy. And they were able to show a benefit to patients within 3 months. So we already believe that there's precedent when you think of showing a benefit in a short period of time. Their benefit is different, of course, than ours will be because the mechanism is different. But the fact that they were able to show an impact on muscle in 3 months is really quite extraordinary when you think of it, because no one else has been able to show that, and we'll be the next. That's our view. And secondly, because we fully expect that we will see a benefit quickly. We saw a benefit, we believe, in 28 days in adults. We saw it in a couple of months in dog. We probably would have seen it earlier in dog if we'd looked earlier. So we think this is a really good way to go. And overall, it will telescope and collapse our timelines. And that will mean we won't, in the fullness of time, need to raise as much money as a company. And then these individuals that are in the 3-month study, again, with regulatory approval, we will roll them into a long-term follow-up of 9 months. And the placebo individuals who didn't receive drug will be rotated into drug. So in that 9-month follow-up, everybody will be on drug. And so this will give us the added benefit of being able to monitor for a longer period of time if there's ongoing and continuing benefit. So this is our plan moving forward. The next slide shows where there will be milestones. And I think what I'll do is just very quickly discuss this and then turn it over to Martin for Q&A. The top line you've seen, we reported out on the phase 1A and the phase 1B, and we still are interested in planning an uplist to NASDAQ later this year. It seems like we keep saying it'll be later this year or early next year, but these markets really are horrible. So we're not going to rush it. We're not going to do anything that will harm the company or, ruin everybody that's tried to support the company. We'll pick our spot as best we can, but we do believe it will unlock further value if we can get there. The 1B that I talked about, we will begin dosing that study. We expect to begin dosing that study in July. We are just in the process of setting up the actual clinical site so that we are at a DMD, essentially a facility that is accustomed to caring for DMD patients, so they have all the right physiotherapy and so on. Then we will have basically a rotation where every 3 months we will have the potential for a data readout. So we'll have data on that later in 2025, early in 2026, and then before the middle of 2026. And then the phase 2, we will begin regulatory submissions in various countries in Q3. These are very different submissions than what we've done for the phase 1. We're preparing those documents now. We're waiting on the completion of some of our studies for the pediatric population that I referred to come together. This is work being done at CROs. So it's just a matter of blocking and tackling now, so to speak. And once we start to get the regulatory approvals, we'll be able to move into, dosing of patients in the second half of the year. Right now, we're in the process of dealing with potential clinical sites. We're not allowed to disclose any of that publicly because we haven't submitted to the FDA yet, so we'll get in trouble if we say too much. But I can tell you that we've identified a number of sites in the US and in other jurisdictions that are very excited about working with us, and we expect to have a lot of support moving forward. And then early in the new year, we'll be able to give updates on recruitment and interim data, and similarly before the middle of the year, updates on interim data, and also moving patients into the long-term follow-up so that everybody can see the progress. So I think this is a really robust approach. I hope you do as well. And Martin, I'm delighted to answer any questions. And if we run a few minutes late, that's fine.
All right. Thanks, Frank. That was a very thorough presentation. Much appreciated. One of the, this is a phase 1B study. The primary endpoint is health, safety, and tolerability. And then you got the extra, you got to look at a few other indicators to see actually what kind of potential benefits there are. Just on a phase 1 basis, safety, you knocked it out of the park, right? There is no issues.
Absolutely. Absolutely.
A lot of companies fail at this. It was a slam dunk.
This is what everybody dreams of, that you basically have, what, for all intents and purposes, what's a pretty clean drug.
Yeah. On the other endpoints, the efficacy endpoints, which we're able to look at, and comparing it with the canines, you weren't able to do a biopsy, or you didn't do a biopsy on the people as you did on the canine samples. Can you discuss that? What the rationale is there?
Yeah, absolutely. So there were two reasons for it. One, individuals at this stage of the disease and this stage of their lives have lost a great deal of muscle. Almost nothing. Well, there is actually nothing known about how much muscle they have left, where it is, literally where it is in their body, and what would be involved in trying to assess it. So we felt, based on advice from our medical advisors and our own feeling, that asking these patients to subject themselves to biopsy, which actually it's a minor operation, but it is an operation, was unreasonable, and would also have a chilling effect on recruitment. And secondly, they're individuals that already are in a lot of pain, and we just felt it would create another barrier that would make this too difficult, and that we wouldn't get very much out of it. That was our prediction. We would not see much because they don't have much to start with, and what are we going to see? So we thought we would do what we could, and we try to see what we can. We believe the reason we saw grip strength is because, over the age of 20 in particular, hands and the ability to use their hands is the last kind of muscle that they have, and the last area where they have some strength. And, I know this anecdotally now from having met many, many, many Duchenne patients, Duchenne adults, and it's very difficult to, I guess, come to the understanding of just how difficult it is for them and how little movement they actually have. So we just didn't think it was the right thing to do.
With the other biomarkers, the serum markers in that, those aren't actually direct indicators. Those are indirect or secondary. They're correlated with muscle and so forth. Yeah. Is it fair to say so? And then there are other drugs in the body interfering with things. So can you just quickly clarify that?
Yeah. So the serum markers that we showed on the slide in the canines were markers of the immune system. All of them on that page are immune system markers. And the cytokines are members of the immune system. So they all have different roles that they play in the immune system. But fundamentally, they respond to the body feeling it's under attack. And IgG that we referred to in a way is kind of the general, or orchestra leader. IgG would only be in muscle, inside the muscle fiber, if the fiber was severely damaged. And then when it's there, it kind of, blows the bugle and says, oh my God, the body's under assault. We've got to come in and clean this up. And then all these, the cytokine cascades and everything else take over. So they respond to muscle. They don't initiate muscle. They're not an indicator that something is starting. They're an indicator after something has begun. And so what is happening in the adults and what happens in children, and the reason that we believe corticosteroids have a benefit is that they knock down the immune system. They knock down all those factors. They suppress them. So that's what we believe is going on. Now, there's huge, huge side effects to that, but we believe that's what's happening. So it's reasonable to us that an adult, after 20 years of corticosteroid therapy, would have an immune system that's pretty tame, which also makes sense in the context that, they're pretty likely to be susceptible to other infectious agents and so on at a greater level than the rest of us, because their immune systems have been suppressed.
I'm curious, with your drug, when I need to get stronger, I need to do exercise. With taking the drug, does physical therapy go along with it, where you take the drug and then need to do some hand or arm exercises to help stimulate it? Or is it, I don't know, spontaneous is the right word, but does it happen on its own, just given the body's mechanism now engaged to build muscle?
Yes. So this is a response to damage. So the body, in response to damage, unleashes a repair and regeneration process. And it's all at the molecular level. It's all at the level of proteins in the body. So if you go to the gym and you are, kind of a super athlete, and you damage your muscles, that will prompt a response of your body to try to repair that damage. And so, we believe that's probably what happens with high-performing athletes. They're damaging tissue. Tissue is then being pushed to repair and regenerate. So the answer to your question, in a way, is yes, but our drug does not need somebody to then go to the gym and exercise, or have a physiotherapist come in and get them walking around the room all day. The body is actually repairing itself. Anything you do in addition to that, we believe would probably be a plus, but it's not a prerequisite. It's the damage itself. And these individuals, their body is constantly, their muscle is constantly damaging, because every time they move is causing damage that you or I would not experience for the same level of activity.
What feedback have you received from the patient community or other research or doctors in the field of DMD? How are they viewing your results?
Yeah, so the response has been overwhelming, quite frankly. Patients are contacting us all the time. Families are contacting us all the time. We were invited by the advocacy group Cure Duchenne to their meeting. I think I mentioned that's the first time we've been at one of their annual meetings. In the past, I don't mean to make any critical comment about Cure Duchenne. In the past, they have been, I would say, at least as a minimum, standoffish about what we were doing, and a wait and see kind of attitude. They didn't leap at this as an idea that they thought could work. And now they've come back and said, look, it's obvious you're making great progress, and our patients are interested. We're interested. We'd like you to come and present. And they gave us a lovely presentation spot and an opportunity to talk to their families. And I mean, we had a lot of feedback, a lot of positive feedback, and a lot of parents coming up and wanting to talk to us. A lot of the patients wanting to come up and talk to us.
All right. I'd like now to talk about the follow-on study. So the initial part of the 1B, what has stopped. The patients have stopped receiving the drug.
Yeah.
But they will then restart taking the drug. I believe in
Correct.
July is the schedule, and they'll then for 11 months from then on see how it continues.
Yes. So we had to complete that first 1B study of 28 days, and then basically say, okay, that's done. Because to go into the long-term follow-up, we needed to submit an update to the regulators in Australia and ask for approval to do an 11-month follow-up study. And so they needed to see the data from the first however many patients. So that's why at some point we had to decide, okay, we have the patients that it's reasonable to expect we're going to get in a short time frame. We have data. It's safe. Let's get it in front of the regulators so we can bring these individuals back and get them on drug for another 11 months. And then we'll, in parallel, we'll ask the regulators, can we now think about bringing other patients in that weren't in the first part, so that if we can increase the numbers, we will.
Okay, so it's going to include the 5 that were part of the
yes.
The one completed.
That's the key.
And you're going to include, if possible, additional patients.
Correct.
As well.
That's our objective. Yeah.
Okay. And just to be clear, because the timing, they're both Q3. This is not your phase 2. This is a continuation of your phase 1B.
Correct. Absolutely.
Okay. Because I got a little mixed up between, I think because of the Q starting in the same quarter.
It got a little. Well, that's a pure coincidence.
Okay. Let's switch on to the phase 2. Do you think we've covered everything, or is there something we're missing in the phase 1B, or do you think we've covered anything we missed?
I can't think of anything. Liz, Anything you think we've missed?
No, I think you covered it very thoroughly.
Okay, thank you. All right.
All right. So with the data from the phase one, the different parts of the phase one, has that changed? Did that change how you wanted to pursue the phase two trial and change that structure around? Because initially you were going to submit an IND, or there's a question here, you were going to submit an IND by the end of Q1 2025, but now not till Q3. Has your strategy changed? And was this as a result of the data you collected to date?
Yeah. So, I mean, the questioner is accurate. That is what we were. That is what we said, and that is what we were planning early in 2025. Was that by the end of the Q1 we would submit something to the FDA, and we were thinking that, and I think we discussed this. I believe we discussed it anyway. Publicly, that we were thinking of moving into a study in adolescent patients, that we would look at boys that were above in their teen years, I guess, and we would do an open label study, so no placebo controlled, so that we could continue to generate data quickly. And as we thought about that more, frankly, and as we started to get glimmers of data from the adult study, we thought that it might make more sense to modify our strategy. Since we had the capital to do this, we thought, if we plan instead to go directly into a placebo-controlled study, where we have that arm that is the comparator arm, and we have 2 treatment groups, and we do that in the population that is essentially the number one target population, which is children, that might accelerate our ability and our timing on getting the drug approved, and potentially have a much greater impact on future partnering or M&A activity. So we did decide to amend our strategy. And that meant that we needed to wait until we had all of the toxicology work done to go into children that are prepubescent. And that basically, all that data comes together later this month, early in July. So that's precisely what we decided to do. It's what we've always felt from day one, is that it's smarter to say we have a better plan. We have it based on information that's coming in. We think this could work better, and to do that. And sometimes it creates a little bit of short-term distress or misunderstanding or something. But I can basically state that we're always trying to figure out the best and the fastest way, without harming the patients or the chance of getting the drug approved. We're trying to figure out the fastest way to generate data that could persuade a regulator to put us on a path to approval.
You've essentially raised the bar on your phase 2 study, going from an open label concept to a placebo-controlled as well. I would think it was initially adolescent. Now you're going to, which I presume there are greater standards for. So, and you've raised the bar. Is that in part because of the strong, like you view this as strong data? It's like, hey, we're getting good data. Let's raise it up. If it works, we're going to be a few steps ahead than the original plan.
Absolutely. I mean, this is what we, inside the company, it's what we all believe. So It is raising the bar, no doubt about it. So how often do companies do that to themselves? But it's because we have confidence. It's not because we've lost our minds. It's because we have confidence in what we're seeing, for what it matters to the audience. We don't think the data that we're seeing is an accident or a fluke. We think it's completely consistent with our understanding of the mechanism of action and our understanding of what we've seen in preclinical models. And we want to go into a population with a study where if we see a difference, it will be absolutely unambiguous. It won't be subject to somebody's judgment or interpretation. It will be subject to statistical data, and where we will be able to sit down with regulators around the world and say, look, we're doing something here no one's ever done.
Would you expect, I don't know how far designed it is, would you expect there to be biopsy data in
yes.
the next step?
Yep.
Okay.
I mean, everything I'm saying is subject to the regulators approving.
Yeah.
But this is what we will ask the regulators for: the permission to do biopsy in the children, to do MRI in the children. And we will do, all of the customary DMD functional measurements, the NSAA, the stair climb, the stride velocity. We will do grip strength. We will do pulmonary function. We will do a wide range of functional measurements. And if anything, we're, highly motivated and encouraged by what we've seen in an adult population.
That you have planned the IND meeting for Q3 of this year. Have you had like a pre-IND meeting or some preliminary meeting to discuss broadly the plan?
Yeah. So just one correction. It's, if I may, it's not an IND meeting in the third quarter. It's an IND submission.
Sorry.
It doesn't need to be a meeting. We submit. And to your second question, yes, we did have a pre-IND meeting, which we had in 2024, in which we, paved the groundwork for this type of opportunity.
And just to reiterate, you said it before, if it goes according to plan, when would the phase 2 begin?
So if all goes according to plan, we expect to have our submission into the regulators in multiple jurisdictions and feedback from the regulators in multiple jurisdictions in Q3, and be in a position to initiate clinical sites in Q3. So access, meaning an approved drug, that's after we get approval. Access, meaning when might we do a clinical trial in older patients, that is not something that we will be able to take on board this year. It's something we will look to take on board in the future after we get the first trial up and running and we make progress in the first trial, the first phase 2 trial I'm referring to.
All right.
It's a population we're very interested in, but one that we now have to, we had to make a choice, and this is the choice that we've decided is the best approach to get the drug approved in the least amount of time so it can benefit the most number of people.
Do you have, or are you planning a compassionate use program?
So absolutely, compassionate use is on our radar, no question about it. It's very important. It's very important to me personally that we're able to do something. We're in a bit of an unusual spot. It seems everything about Satellos some days is unusual compared to everything else around. But we're not a US company. We're a Canadian company. And compassionate use is an American phenomenon. So exactly how we make that happen is a little bit unclear at the moment. We cannot do it until we're actually in the US with an active clinical trial that's up and running. So as soon as we have that in place, then we will start looking at how best to undertake a compassionate use program in the US. I'm not aware of there being an equivalent in Canada. So at least for the time being, it's to us pretty much a US phenomenon.
I think I've addressed the questions from the audience on sort of the medical scientific side of it. There we have some on the more on the business side. Before we, let's say, wrap up the science side and the trial side, are there any other comments you want to make regarding that?
I'm good, Martin. Thank you. I think the questions have been really helpful to learn what's on the mind of the audience. And also, I think we've had a good discussion. So I'm good. And if you have questions for Liz, I would be delighted.
There are several questions here, especially given what appears to be very solid data coming out of a phase 1b trial about frustration or disappointment with the share price. I don't know, any comments you can say to that?
Well, we're frustrated and disappointed with the share price as well. It's something that confuses us. We don't know exactly what is happening. It's been a very difficult market overall. It's been a very difficult biotech market, particularly the last couple of months with all the changes at the FDA, the tariffs, interest rates, all those kinds of things have been having a lot of impact on biotech stocks in general, and it seems to some of it is flowing into Satellos as well. We are, working very hard to talk to as many existing investors and new investors as possible, to focus on finding new buyers to help stabilize and hopefully improve the stock. But we do believe the value is there. We've added value with this phase 1b clinical data, which we're really excited about, and the market will realize it in time. We'll do our best to talk about it as often as we can.
Because normally with biotech skewing, strong safety is a huge de-risking event, and that sort of commensurate share price increase, maybe a little too many expectations in investors' minds on what kind of data could get presented there.
There's definitely a disconnect happening. One of our competitor companies had data this morning, which we thought was quite incredible, and their stock was down somewhere between 15 and 20% today. So it's hard sometimes to understand exactly what the market is looking for.
What news events could be catalysts for the next coming up while? I guess submitting your package, the starting of the continuation of the phase 1b. Talk about some of the catalysts that potentially could be, or we expect to be coming up over the next little while.
So we do think that getting the IND cleared with the FDA will be an important catalyst for a lot of investors. We all know the FDA is considered to be the gold standard when it comes to regulatory. So clearing that hurdle, knowing that there's no concerns and that we can move full speed ahead in the phase 2 is important. Getting that first patient into the study and knowing that data is coming. It's a short study. It's a 3-month study. So those are near-term important milestones. One of the things that we're really excited about is the phase 1b long-term follow-up. So those 5 patients that we've already dosed will have the opportunity to come back into the study, continue to get drug for an additional 11 months, and we'll continue to monitor those patients every 3 months. So we'll continue to look at their grip strength. We'll look at some additional measures like MRI, for example. And that's open label data, so we'll be able to see it real time as it comes in. And what we know from our animal studies are that the longer the animals get the drug, the stronger they become. So we're hopeful that we'll continue to see improvements in those adult patients, which would be really exciting in that patient population, and also really exciting because it's data we'll be able to share on a regular cadence. So that'll be the first kind of look at that will be kind of late Q3 or early Q4 as well. So that could potentially be a really important catalyst for the company.
So just to be clear, for the ongoing 1B trial, we're not going to have to wait the 11 months to get the data on those 3-month interim data. You'll be able to release some top line data on that?
Yes, exactly. Yes. Exactly.
Okay. All right. With the potential uplisting to NASDAQ, and then as well, the questions here on potential share consolidation going forward. Are you waiting for there to be hot markets before going to NASDAQ, getting some good data? I know your thought process and what needs to come together for that to happen.
Yeah. So we believe that in order to really get the value the company deserves, when we reveal the data from the phase 2 blinded placebo-controlled clinical study, we need to be on NASDAQ. So between now and then, we need to figure out when the right time is. So a lot of shareholders know that we asked for approval to do a reverse split this time last year, which we got overwhelming support from our shareholders to do that. But we made the decision as a management team that it was not the right time to do that. So we pushed that off. And so we're asking for approval again for another year. But it's something we take very seriously. We worked very diligently around the financing in December, and we were really pleased to be able to raise capital at 90 cents, where just weeks before we had been trading at 50 cents. It was a straight common deal. We did not issue any warrants. It was very clean, placed with a fantastic syndicate of US institutional investors. So when we think ahead to the reverse split, we'll be very thoughtful about the appropriate ratio and the appropriate time. So we want it to be seen as a building of momentum towards a listing. So it's likely going to be in conjunction with some data, and then in conjunction with the listing itself. And so often with reverse splits, 98% of the time you see a reverse split, a company's doing it to try to maintain a listing. So their share price has dropped, they're below $1, they have to do a reverse split in order to keep their NASDAQ listing. And so it's often after not the best news, the reverse split is happening, which leads to further depreciation in the share price. But we do see examples when a company is doing it as part of a momentum build towards a listing, the outcome is very different. So we've always looked at Bellis Health as a great example of a company that did a reverse split to list on NASDAQ, and the share price maintained initially, and then of course grew significantly over the next couple of years. So that's how we will work to build for Satellos, getting ready for that NASDAQ listing.
Have you had any partnership talks with any big pharma or other pharma companies? And at what phase, like you're still fairly early stage, at what point would it be reasonable to expect that could be a possibility? Is it too soon to potentially announce something like that, or would that you need additional data for reasonable expectation of something like that?
We do have a lot of interest right now, and it's been building. So despite the conversation we just had about the market and stock price, companies that are in the field have been following us, talking to us, reaching out to us. So those dialogues are beginning, and I'm not going to promise anything at the moment. It's not my style or our style, and we're not at the moment thinking that where we are in terms of market cap is where we want to start thinking about a partnership or an M&A. But over the next, literally, we've just discussed it. We will be starting a phase 2 trial this year. We will be conducting a long-term follow-up study in adults starting this year. We will be reporting data from those 2 studies on an ongoing basis, starting probably end of Q3, beginning of Q4, in the 1B long-term follow-up, and then every 3 months after that between those 2 studies for several quarters. So if none of that drives value, then obviously we're in a very strange world. But, our view is what we can control is setting up to do the absolute best we can to get this drug approved. And along that path, generating data that says we're helping the patient community or we have significant potential to help the patient community is what's going to get us either to NASDAQ or bought out or both.
We've gone well over time here. I very much appreciate giving us so much of your time. Any final comments or thoughts before we wrap it up?
I think my last comments were probably, how I mostly feel about the situation where, as we've said, and as I've said before, we're doing something nobody else has ever tried to do, which is to regenerate human tissue inside the body. If we're successful in doing this, it will be a first. That is going to change medical practice. I mean, that's how we look at what we're doing. And we're starting with Duchenne muscular dystrophy. It is radically different. Why it's taking the time it's taking for value to be recognized through the stock market, as Liz said, kind of mystifies us most of the time. It's not that we're not working at it. It's not that we haven't hired advisors. It's not that we're not doing outreach. We're doing all of those things. And I think it's just more data, the right kind of data, and the data that is unambiguous, that nobody can look at the data. What we need to get to is a point where no one can look at the data and say, oh, somehow you played games with the statistics, or somehow you're telling us this, but you're not telling us something else. Because we threaten a lot of companies. I mean, frankly, we threaten a lot of players in this space and in other spaces who have been working on medicines for a long time that aren't showing the kind of functional benefit that everybody hoped for. So, stick with us because, we're not giving up.
All right. And yeah, you've gotten great data so far. So it's all going in the right direction, if I may editorialize here. Thank you very much. I really appreciate your time. Very informative. Thank you, audience, for your patience and staying through this long presentation and digesting all sorts of information here. Have a great day, and looking forward to our next meeting.
Thank you, Martin. Bye now.
Cheers.